हेलो फ्रेंड्स, चलिए आज हम स्टार्ट करते हैं क्लास 11th का सेल साइकिल एंड सेल डिवीजन। इस चैप्टर में हम जानेंगे कि कैसे सेल अपनी साइकिल को कंप्लीट करता है और कैसे डिवाइड होता है। तो चलिए स्टार्ट करते हैं सेल साइकिल एंड सेल डिवीजन। As we know, growth and reproduction are characteristic of living organism and cell. As division, there is a single cell which divide to form two cells, and that after it grows, देन आफ्टर दैट इट फिर कंटिन्यूसली ये डिवाइड करता है, ऐसे ये डिवीजन कंटिन्यूसली चलती रहती है। तो देयर इज अ सेल पॉपुलेशन कंसिस्ट ऑफ मिलियन ऑफ सेल्स। नाउ सेल साइकिल it is a sequence of event by which cell replicate its genome, grow and divide to form daughter cells. Now, cell growth. Cell growth, it is a continuous process throughout cell cycle, and genome replicate in S phase. It is very important. Event of cell cycle are undergo genetic control. Now we can talk about phases of cell cycle. There is a there are two phases. One is interphase and other is M phase or mitosis phase. तो चलिए इंटरफेस के बारे में जानेंगे. It is previously called resting phase because it is a preparatory or division phase. In this phase, it is a phase between two successive division and 95% duration of cell cycle. It divided into three parts. First G1, second S, and third is G2. Now we talk about G1 phase, or we can also call it Gabon phase. It is the interval between cell division and initiation of DNA replication. It is metabolically active cell, and cell organelle are duplicate here, and it is continuous growth. Now, S phase or synthesis phase. Here, DNA replication occur in nucleus and chromosome replication या duplication. सॉरी, यहाँ पे क्रोमोसोम की स्पेलिंग मिस्टेक है, प्लीज करेक्ट इट. इन एनिमल सेल्स सेंट्रियोल डुप्लीकेशन अकर एंड नंबर ऑफ डीएनए डबल, but the number of chromosome remains same during this stage. Now Z2 phase या gap 2 phase. Here protein for mitosis or synthesis, that is important, and cytoplasmic growth occur. Now we talk about M phase or mitosis phase. Here, it is the actual phase of cell division, and it can be divided into two parts. First is karyokinesis and other is cytokinesis. Karyokinesis, it is a nuclear division, and cytokinesis, it is a division of cytoplasm. Here we can see it is a G1 phase, and this is S phase, and this is Z2, and this is M phase. Now, some important point. Duration of cell cycle vary from organism to organism and cell type to cell type. And duration of cell cycle in human cell in culture is about 24 hours, and duration of cell cycle in yeast is about 90 minutes. Mitosis in animal occur usually in somatic cell or 2N cell. There are few exceptions where haploid cells are divided by mitosis in animals. For example, male honeybee and mitosis in plant, that is N or 2N cells. Now we talk about G0 phase. It is a phase which come after the G1 phase. In adult organisms, some cells do not divide and exist from G1 stage and enter in quiescent phase. For example, heart cell. It mainly suspended the cell cycle and there is no cell division. And cells are metabolically active and enter the G0 phase at the end of G1 phase. Now we talk about M phase. Here, M phase divided into two phases, that is karyokinesis and other is cytokinesis. Karyokinesis it divided into four phases. That one is prophase, then metaphase, then anaphase, and telophase. Here we talk about further in complete detail. Now, M phase. It is the most dramatic period of the cell cycle. This line is very important and major reorganization of virtually all component of cell. Here, mitotic division is equational division because number of chromosome in parental and daughter cell are same. And mitosis start with karyokinesis and end with cytokinesis. Cell division is progressive process. Now, prophase. It is the initiation of condensation of chromosomal material, and chromosomal material, या chromatin, undergo untangle, that is chromosome. Now, movement of centrosome toward opposite pole. Formation of ester and mitotic apparatus. This is very important because this line come in neat as direct. And mitotic apparatus equal to two ester plus spindle fiber. At the end of prophase, nuclear membrane, nucleolus, and endoplasmic reticulum and Golgi body disappear. Now we talk about metaphase. That is onset of metaphase, complete disintegration of nuclear membrane, and there is condensation of chromosome material completed. It is the best stage to study morphology of chromosome and spindle fiber attached to kinetochore of chromosome, and chromosome arranged on equator of cell and form equatorial plate. Now we talk about anaphase. Simultaneous selecting of chromosome, that is onset of anaphase, and each sister chromatid become daughter chromosome. And pole-wide migration of daughter chromosome occur, and there is centromere is toward pole, thus forming leading edge and arm are trailing behind. And now we talk about telophase. Here, chromosome cluster at opposite pole, and chromosome decondensation occur, and there is chromosome lost their individuality, formation of nuclear membrane around chromosome cluster, and there is nucleolus, Golgi body, and endoplasmic reticulum reform. Now we talk about cytokinesis. It is the formation of two daughter cells by separation of cytoplasm. Here, distribution of cytoplasmic organelle in daughter cell. Cytokinesis in animal cell, or we can say animal cell cytokinesis. Here, appearance of furrow in plasma membrane and furrow deepens and joining in the center and cell divide into two cell. And there is cytokinesis in plant cell. There cell plate and joining in the format or a simple precursor cell plate. Cells or cell become middle lamella plate a simple pre-Golgi cell. When karyokinesis is not followed by cytokinesis, it is a result of multinucleated condition, that is liquid endosperm in coconut. Now we talk about significance of mitosis. Here, production of genetically identical daughter cell occur, growth of multicellular organism, and restoration of nucleocytoplasmic ratio. Here, cell repair, rearrangement of dead and damaged cell. तो यहाँ पे कुछ स्पेलिंग मिस्टेक है, प्लीज आप उनको सही कर लीजिए. Here, continuous growth of plant by division in apical meristem and lateral meristem. Now, we talk about meiosis. Meiosis, it is a specialized type of cell division which reduce number of chromosome to half of daughter cell, that is here 2n. After meiosis, it is haploid, that is n, and after fertilization, it is again 2n, that is diploid phase. Now, meiosis involves two sequential cycle of nuclear division and cell division, but only a single cycle of DNA replication. This is important. And meiosis involves pairing of homologous chromosome and crossing over. Here, meiosis is divided into two phases, that is meiosis 1 and meiosis 2. In between meiosis 1 and meiosis 2, there is a phase that is interkinesis, and meiosis 1 divided into G1S and G. Now we talk about meiosis 1. Meiosis 1 का prophase 1, chromatin fiber are colored in shade of hue and age with small emerging chromosomal condensation. Now we talk about zygote. Zygote, homologous chromosome are clearly paired. One homolog is deep below and chromosome are gradually visible. Now zygote, it is a pairing of homologous chromosome, that is synapses. And now we talk about vegetin. The pairing homologous chromosome are blue-green color and thicker. Vegetin bivalent are clearly appear as tetrad and recombination nodule formation and crossing over between non-sister chromatid, that is chiasmata of homologous chromosome. Now recombinase enzyme activities are here in vegetin. And now we talk about diplotin. Here dissolution of syneptonymal complex occur and chiasmata formation. And diplotin can last for few days to many years in oocytes of some vertebrate. Now, dikinesis. It is a transition to metaphase 1. Here, condensation of chromosome material complete and mineralization of chiasmata membrane disintegrate. Now, metaphase 1, that is, and bivalent are arranged on equator metaphasic plate, that is, spindle fiber attached to the kinetochore. Anaphase 1, separation of homologous chromosome and poleward movement of chromosome. Sister chromatid remain associated at their centromere. Now, telophase 1, here nuclear membrane and nucleolus reformation. Now, some important points. Some dispersion of chromosome may occur, but they do not reach up to fully extended stage. And cytokinesis and formation of diet of cell. Interkinesis, it is a stage between meiosis 1 and meiosis 2. Here, DNA replication does not occur. It is short duration phase. Now we talk about meiosis 2, that is similar to mitosis. In prophase 2, nuclear membrane disappear and chromosome become fully compact. And metaphase 2, chromosome align on equator and spindle get attached to kinetochore. Now anaphase 2, splitting of centromere and pole bar movement of daughter chromosome by shortening of microtubule or spindle fiber. And telophase 2, nuclear membrane reappear, that is cytokinesis and formation of tetrad of cell. Significance of meiosis here: conservation of constant number of chromosomes in sexually reproducing organisms across the generation and increased genetic viability in population. Here we can see meiosis 1 and meiosis 2. That is meiosis 2 is like a mitosis. So thank you.